This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, a special episode recorded on August 17, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. I'm back in Ithaca, New York at Cornell University. And my guest coming back for a second time is Gary Whittaker. Welcome back.
Okay, thank you, Vincent.
You were on TWiV 436.
Yeah.
And this is going to be in the 900s.
Wow, it's time flies. 5 years ago, is that right? Approximately.
Let's take a look. I think it was 2017, yeah. And a lot of things have happened in the meanwhile.
Yeah, absolutely.
And so I know that you were always been a coronavirus virologist, right?
Not always, no. I joined fairly late, and actually, well, after SARS.
SARS-1.
SARS-1,
okay.
Yeah. So before that, it was influenza. It was herpes and then influenza, and then I became a coronavirologist, after SARS-1, and then the field embraced me, and I I loved it, so I stayed.
But your interest had always been spike.
It's always been
right?
viral glycoproteins. Everything started with equine herpes virus for my PhD, understanding herpes virus glycoproteins, and that was too complicated.
Right.
And then I kind of made a detour to influenza, which was more to do with nuclear import and export. I went to Yale to Ari Helenius and did my postdoc there just to learn some cell biology, and then kind of came back to HA when I got to Cornell. After a couple of years talking to Colin Parrish and endocytosis, and that really got me back to HA and viral entry at that point.
Yeah, you have been working on coronavirus longer than many people who started during the pandemic. And you also have an appreciation for viral glycoproteins. So that's what I wanted to talk a bit about today, and also your interest in feline coronaviruses. Could I see that big virus? Is that a coronavirus?
That's a big giant SARS-CoV-2.
Look at that! This is awesome. This is giant microbes, right?
Yeah.
Yeah.
I work for the giant version.
So they have a gigantic...
They do have a giant SARS-CoV-2.
But look, they say COVID-19, then in parentheses SARS-CoV-2.
I know.
Can't they call it the right thing?
Yeah. Well, most people call it COVID. Very few people call it SARS-CoV-2 out there in the world. Which is interesting.
I don't get it because it's like HIV AIDS,
HIV
right?
AIDS, yeah.
It's two names,
right.
Virus and a disease. I don't get it, but I think it's a losing battle, so I'm not going to fight it. Anyway, I wanted to talk about the spike a bit. So can you just review for us the function of the spike in entry and what has to happen for it to bind and catalyze fusion of SARS-CoV-2?
Right. So with SARS-CoV-2, then we've got this, the spike protein here in yellow. That's small, a little undersized, but it's a big, complicated glycoprotein. So that was the realization when I started is you think about influenza HA, which is small and compact, right. And then SARS and this coronavirus spike is just giant and complicated in comparison. So that was it's fundamentally the same things going on, but just more activity, more regulation, more of everything.
Is that a feature of all the corona spikes? They're all big?
They're all big. Yeah, they're all big.
So in that model, there's not many of them. Is that also a sparse?
Relatively sparse. I mean,
much less than flu.
HA is pretty much packed. Yeah. It's not like HIV, which is really sparse, but I think it's somewhere in the middle.
Okay.
So it's the receptor binding. It's the major antigenic component. Although it's very heavily glycosylated, so that that gets shielded a lot. But that's the surface component. It binds the receptor, which we all know now is ACE2, mm-hmm. and then it catalyzes membrane fusion. So what we've been always focused on is like that transition between binding and fusion. Really how does that information get transferred? And a lot of it comes down to these proteolytic cleavage sites. So this is the regulation. So there's a binding to ACE2, and then either before or after there's a cleavage. And in SARS-CoV-2, then it's the famous furin cleavage site, which is not a term I particularly like, but that's what it gets called. And it is a furin cleavage site, but it's not the best terminology, I don't think. Partly because it's, as many people now know, it's not it's certainly not an ideal furin cleavage site sequence-wise. It's weird. It's got some arginines in the wrong place, and it's clearly changing itself. So it's weird as a furin cleavage site, but it's there, and it does get cleaved by furin. But it could well get cleaved by other proteases too. That's what people don't really. Just because there's a furin cleavage site doesn't mean it has to get cleaved by furin. There's lots of other proteases that could come in. Temperase-2 can actually cleave that very well, and as well as a lot of furin family members.
So there are on the SARS-CoV-2 spike, there are two cleavage sites, right? There's S1, S2,
and then down in S2 prime is down in S2 is then S2 prime, that's the one that's linked to the fusion peptide.
So that has to be cleaved to expose fusion, right?
So going back to the HA analogy, that's if you look at influenza HA, that's stripped down. So it's got the cleavage site, it's got the fusion peptide like side by side. But then the coronaviruses add on this optional S1, S2 side as like an extra layer of regulation. So it's the S1, S2 is entirely optional. I would say approximately it's about a 50-50 call whether it has it or not for a coronavirus.
So for SARS-CoV-2, if you block the S1, S2 cleavage site, you don't get fusion?
Yeah, you don't get fusion. Yeah.
So what cleaves S1, S2?
What cleaves S1, S2 in SARS-CoV-2 is it can get cleaved by furin, probably does get cleaved by furin in many cases.
And these are intracellular?
These are intracellular, so it's on the way out of the cell. So furin is typically in the trans-Golgi network, so it's getting cleaved on the way out. And then it's sort of primed at that point. So I call that priming or pre-priming, and then the activation is then on the way in the next cell.
So that's
which is the S2 prime.
S2 prime. So that can be cleaved at the cell surface.
Yeah, or in the endosome.
Or in the endosome.
Right.
And the cell surface would be temperase-2 and related proteases, right?
Right. And then in the endosome, it's cathepsins or cathepsin-related proteases.
So if you change the furin site, which is upstream, then entry is very much made less efficient, right?
Well, yes. I mean, it's very context dependent. This is the problem. It's very context dependent. So actually, we did some studies early on where we blocked the furin cleavage site with an inhibitor, and we got much better pseudoparticle infection. It would actually increase the cleavage. Pseudoparticles, yeah. Okay. So we have to discriminate between like actual virus and pseudoparticles, which is it's easier to overlook that. I mean, and one of my colleagues, Nihal Anton-Benet, has got this model of secretory lysosomes. So the pseudoparticle is actually going out of the cell in a very different pathway to the live virus, probably. So we have to be very, very careful not to, think too simplistically and not assume that it's the same. And that's a challenge.
So you have an inhibitor, but you only use that against pseudoviruses.
Yeah, we at the time we were still getting all our BSL-3 stuff together. This was very early on in the pandemic, so we could go straight to pseudoparticles while we were waiting for the CDC to deliver the virus as much as anything else.
So that inhibitor, which is N something, is it?
Just the standard furin inhibitor. A peptide inhibitor.
It's a furin inhibitor. It's not a temperase inhibitor. All right. So the furin inhibitor makes pseudoparticles more infectious.
It makes them more stable, I think, is what happens. So that's what the virus is sort of in this sort of zone of infectivity and stability, which is when I talk a lot about viruses when I teach students, I say it's this sort of there's a stable particle that suddenly becomes unstable when it needs to get into the cell. So it's in this area. So once the S1, S2 side is cleaved, then that particle gets pretty unstable. It flips up in many cases, the RBD to make it find the receptor more efficiently. But in the process, it's making it fairly unstable. So it's sort of pushing that window of like between stability and instability, and it works enough to be highly transmissible. But it's not kind of, if virus want to do anything, it's not what the virus wants to do. It wants to be like SARS-1 and be nice and stable and just sort of happily sit in a virocell and get on with its life.
But if you change, others have changed the furin site, right? Yeah. And that reduces infectivity and pathogenicity in animals,
so
right?
It does. I mean, it clearly reduces transmissibility. I think that's the pathogenicity aspect is a little less clear, but certainly I think it's clearly a transmission marker, if that's the right word, to allow it to become more transmissible.
I mean, this furin site. What's a better name in your?
I'm coming down to S1, S2 loop. I'm not sure.
S1, S2 loop.
Yeah, so it's a structural loop that's between S1 and S2, and that's what I'm that's my sweet spot right now is S1, S2 loop.
So as you know, we talked about yesterday, many people thought that the presence of it made it certain that it was made in the laboratory.
Yeah, right. Yeah.
But is that because no other beta coronavirus?
No other beta coronavirus has it. Yeah. So
that we know of, right?
That we know of, right? So that's the question. Is there's almost certainly lots of beta coronavirus we've never sampled, so we don't know it. We just don't know that they're.
I remember there's one bat coronavirus recently sampled that has there's some activity. There's a lot of activity, sort of genetic activity selection, if that's the right word, in that area. So you can start to see loops coming and going, but it's the combination of the loop and the basic residues which create that furin cleavage site. And other coronas, not betas, but other coronas have furin cleavage.
Yeah, right.
Not SARS-CoV-1 though, because that's a beta.
Not SARS-CoV-1, but MERS has furin cleavage sites. A lot of the MERS, HQ5, for instance, has sites.
And now your feline corona.
My feline, yeah. So that's
yours,
mine. So this is. I don't want to claim ownership of the virus, but this is. I think a really interesting scenario to think about. So it's an alpha coronavirus, right? When we very first started with. I've still got the slide somewhere on my computer about cleaved or uncleaved. So the alpha coronaviruses were uncleaved spikes, and the gamma coronaviruses had cleaved spikes. So gamma is infectious bronchitis of chickens. So that's the prototype coronavirus isolated in 1940 or so.
Right.
So gamma coronaviruses are always cleaved as far as we know. Although that's probably not quite accurate now, but alphas are not cleaved at all, except with the one big exception is like what we call feline coronavirus 1. Mm-hmm. So that if you did that alignment, you'd see a big gap, and then you'd see like a furin cleavage site sitting like right in feline and canine coronavirus 1. So that virus certainly wasn't, that wasn't a lab leak. That wasn't natural. It was it was first identified in. Well, the disease in 1963, I believe, and then the virus in like the 1970s. So it's been around for 50 years, and the origin of that is very mysterious. So if anybody out there in TWiV land wants to think about that, where did this cat virus come from? That's a very interesting question to me, because its origin is just this outlier. It's a complete outlier alpha coronavirus.
And there's nothing similar to it. That would nothing in bats or anything.
Well, no, we don't. There's no bat origin. There's no rodent origin. It's just sitting there out. It's very closely related to let's say the traditional feline coronavirus is what we call type 2. So if you go to ATCC and type in FIPV, which is the virus, you'll get the type 2 viruses. So they're like very. A lot of virologists study those. They're very convenient viruses to study. They grow in cell culture. They behave very well. Yeah. So that's why ATCC you can order it from ATCC. But the type 1 viruses, it's like noroviruses or something. There's no cell culture model, so we can't grow them in the lab. So this is why they're very understudied. They're an enormous problem in cats, but we can't grow them in the lab. And I really think the reason for that is because of this cleavage site. It makes the protein quite unstable. And we haven't been able to do the vero cell trick of getting it to adapt and infect a vero cell and switch to a different pathway. Although that's something we should probably come back and think about more, because it's there's the other story I like to tell people is OC43 is actually there's actually a point mutant right in the cleavage site of the ATCC strain of OC43, and it doesn't match the clinical strains. It's been selected to through that cleavage site to grow in cell culture.
So if you pass SARS-CoV-2 in viros, you lose
lose it very rapidly. Little deletions and point mutants come up the 3 passes so we got out going back 2.5 years to the start of the pandemic. We were on the list to get the Washington 1 strain from CDC right away, but pretty rapidly they were sending it out with deleted furin cleavage sites
because they grew it in
vero cells and didn't realize that.
So viros don't have cell surface temperance.
They don't have cell surface temperance, and now the standard is just to grow that in vero temperance 2 cells to maintain that cleavage site.
So that what happens in viros without temperance is the virus goes into the endosome where
it just switches to that endosome pathway.
So why is the furin site lost so quickly then?
I think it's because it makes the particle relatively unstable, and that's not the virus doesn't like that. If viruses like things, it's not a happy virus.
It's not fit, right?
Yeah, it's not a very fit virus.
Okay, and just a few passages. So, and viros are kidney cells from monkeys. So,
right.
I mean, I don't know about human kidney. Does human kidney have temperus?
I don't know. I mean, the whole protease world is a big black box. When you start digging around and like figuring out what protease is in what tissue, it's very murky, and and then it's not just temperus 2; it's temperus 1 through 14 or whatever. There's a whole lot of temperuses out there.
So the moral of the virus story is that there's never any one answer. There are multiple.
Yeah, there's always multiple answers, and coronaviruses take advantage of that like big time. They
Journalists don't like that.
Yeah, no.
So let me go back to the spike. It's a trimer, right?
It's a trimer.
And the head, the RBD can go up or down, correct? And to bind it has to be up, right?
Well, it binds better when it's up. I think it can still bind when it's down, but it's just much more efficient when it's up.
Do all three have to be up to bind, or no?
Well, I think the more up, the better binding, but the more up, the more unstable. So it's, but I think...
And to get up, it needs the furin cleavage.
The furin cleavage site kind of helps it.
It's now SARS-CoV-1, which does not have a furin cleavage. Does it have this up-down conformation?
I don't know if anybody's really... It's possible that's been studied, but I don't know. But I think the general idea is they're often very sort of dynamic structures, but whether that same up-down flip is... I'd have to turn to my structural biology colleagues to kind of figure that one out. But in principle, it's in a down conformation, so it still binds, but not as well, and that's why it's not really hitting those low H2 cells probably in the upper airways, and that's why it's not really able to transmit as well. It's
because it goes down further into the lung.
It needs to get further down where there's more H2 around.
So in the course of the pandemic, as variants have arisen, have any lost the furin site?
They've actually gained furin cleavage. That's the kind of... So this is where it starts to look. So Delta is like what happened, it was, there was 4 amino acid insert, PRRA, right, which... So that first of all, that proline is weird. So the R is in the wrong place and the proline is there. So that just makes it unusual.
And by the way, people use that as an argument for human engineering, but a human wouldn't make those mistakes.
Well, if you did, I mean, the thing is, if you wanted to do it, you wouldn't put that sequence in. You would put something different in.
But why would you even put a furin site in? Would there be any precedent for that?
Well, people have done that. I mean, just to see what happens. I mean, we've done it in not in a live virus, but in a
like
just
a protein,
like a protein. Just that was part of that just helped. Back in 2009, when we published on the S2 prime, we identified S2 prime partly it actually goes back to IBV Baudet. So IBV had a strain, back to the scenario, we want to study the virus, but we can't grow it, right? Mm-hmm. So we're kind of stuck as virologists. So IBV, again, doesn't grow well in cell culture, except for this one strain, which is called IBV Baudet, which was selected way back in 1940 something as a vaccine strain. So they took it and put it through 100 times or something through chick embryos and made this virus that now infects BHK cells really nicely. So we went right to that. And we saw, and then if you look at the sequence, then you'll see there's a furin cleavage. There's actually 2 furin cleavage sites at those 2 positions.
Okay.
So that was what was the sort of the little light bulb went off, and I said, okay, that's what's going on. The first position was known about. The second one wasn't. It's only present in IBV Baudet. But that was the sort of. Then if you go to look at all the other IBV strains, you'll see that there's arginines there, but they're just not cleaved. They're still cleaved, but not through furin. It's like the temperus 2 site, and they're just a lot harder to spot through the sort of basic sequence gazing, because you're just looking for that little run of basic residues, which sort of pops right out. So Delta has an improved furin. So we've got the proline. So the proline went to histidine in the alpha-like viruses, and then histidine went to arginine. So now we've got an extra arginine, which improves the cleavability by furin. So it's actually more cleaved, more fusogenic, and more transmissible, presumably. And then with Omicron, we've kind of everything's gone a little weird. So we lost the arginine went back to histidine, but we added an extra lysine, a little 2 amino acids away. So we've kind of got a new furin cleavage site. I have a little debate right now with some colleagues out in Davis, physics lab in Davis, who contacted me because we published that that was actually increased we do a lot of work with biochemistry, with peptides, and that was in our study that was increased. So Omicron was the highest cleaving virus, and the argument from the physics lab is they've done modeling and said you're not correct because the peptides are in the wrong orientation in the furin binding pocket.
But you did the actual experiment.
So, right, we did the experiment, but this is in solution, so the peptide can go in this way or this way. It doesn't care, but on the context of a spike, it's locked into place. So the modeling actually said what we measured was actually in the wrong orientation.
So you didn't look at virus infectivity, in other words?
Not yet, no. We're just looking at pseudoparticles. Because we're looking at peptides. We're moving up. We're trying to get pseudoparticles and things like that. It takes time to get all this stuff done. The peptides are kind of fairly quick to do.
But some have said that Omicron is no longer entering via the surface. It's going through endosome. Is that correct?
Yeah, I don't think that's correct. So I think maybe BA1 did a little detour, but BA5, let's say BA5's come back. So I think that the very first papers were saying it was skipping TMPRSS2 and going straight to the endosome. I think that was, again, another oversimplification.
I mean, it's all done in cell culture, right?
It shifted, right? It definitely shifted, but it sort of shifted back. And that's where, as some of your audience may know, that we've got this new COVID drug, like N385, which is a TMPRSS2 inhibitor. And that was the big fear. Like we've got this drug that works great up until Delta, and then all of a sudden it's not going to work. But it turns out it works great, and the drug works just fine on Omicron. So I think that story is, again, it was one of those oversimplifications.
Is that drug going into people?
Not yet. We're trying to get funding. So one of the things I'm trying to do right now is get some bridge funding from the NIH to kind of move this forward.
All right. So to make clear, both Delta and Omicrons, all the Omicrons are nicely inhibited by a TMPRSS2 inhibitor.
Right. TMPRSS2 inhibitor really shuts everything down.
And what kind of cells are you doing this?
Well, we've done it in Cali-3 cells, range of organoids in mice. My colleague Hector here is Hector Aguiar-Carreno has put everything into mice, and it works really well in mice. We're just moving to hamsters and ferrets and everything else now, but it works great on. It was developed.
Don't move too far. You're going to go out of range. Come a little closer.
My legs here. It was actually started out as a influenza drug. Actually, so we partnered with a lab in Quebec at Sherbrooke, and Richard Leduc is the guy there, and it was one of those. I got lots of, at the start of the pandemic, I got lots of emails saying, hey, can we work on SARS-CoV-2 together?
Yes.
And I said yes to a lot of people. Unfortunately, I couldn't say yes to everybody, but I definitely said yes to Richard because I know he's a protease expert. He really understands proteases really well. And he had a medicinal chemist up there, and they'd got these drugs, these peptides, small molecule peptides, which were blocking influenza really well, and they wanted to see if they worked on SARS-CoV-2. And so we said sure, and everything worked out really well. So that we've got that published now. We're just trying to get all the funding together to kind of move everything forward.
Why does it inhibit? Is it a influenza with a furin cleavage site?
No, it's a TMPRSS2 inhibitor. So it's a TMPRSS2 cleave. That's the protease.
That is the protease that cleaves HA.
Cleaves HA. Yeah. So it's not related to furin at all. It's just so.
Okay, so if you have a high path H5, that's furin cleavage.
That's furin, yeah.
Okay, so that's different.
So that's different. So we haven't actually checked that. We're assuming it doesn't. We don't. That's the select agent, so we can't do that experiment. But I'm assuming not. But right now we're checking it out against H7N9, for instance, and that's underway right now.
So the changes in the furin cleavage site from the original isolate through Delta and Omicron probably make no difference in human infections.
I don't know about no difference. I mean, that's again.
But the things you see in cell culture are exaggerations, maybe.
Maybe. Well, they're oversimplifications. If that's
so. This is the conundrum,
right?
In cell cultures, it's a model. It's not people.
It's not people.
How do you find out what happens? You can't.
Really. You need to. I mean, if it was influenza, you go to a ferret, which is like the closest to a person where you can do an experiment. I don't think SARS-CoV-2 in a ferret is not a very good model. With the infant, you go to like the best animal model you can find. I guess probably you go to non-human primates, but that's it's
hard to do.
It's hard to do. So yeah.
So that's the part of the problem. There's no really good animal model that's convenient.
Right. And then with Omicron, it's just got worse with Omicron because then Omicron's just like not none of the animal models work. I mean, we did all our work with the K18 mice, because that's what was available, and they're a great sort of severe model. But again, they don't really model disease very well. I mean, it goes to the brain really easily, and a lot of coronaviruses go to the brain with these mice really easily. So it's not, they die of encephalitis, not of pneumonia, for instance. So it's just,
and that's not what happens in people,
right?
Right. Well, I always have issues with conclusions made in cell culture and saying this is what's happening in people, but you can't always say that. I
mean, it might be a good model, it might not. You just have to kind of be aware of the limitations and do what you can. And ultimately, you've got to go to public health and do things in people, and that's but that gets messy very rapidly.
So, I mean, I can see variants changing to evade antibody, right?
Yeah.
We know that from influenza virus, right? But I don't understand these furin cleavage site changes. And then there are changes in ACE2 binding, which get higher and then they get lower. And it's right now the virus it's new. It's a new virus. It's in this new population. It's just sort of sampling its territory to try to figure out where it needs to settle. Eventually it will settle. It's just taking a heck of a long time to settle and causing a whole lot of grief along the way. It's way more than I think anybody. I mean, I'm supposed to be a coronavirus expert. This is like way beyond Eddie. I thought it would settle in it'd be like a flu pandemic kind of. It will settle. Give it a few months or a year, it will settle. But it's so is the extent of variation surprising to you?
Yeah, it's surprising. Yeah, and I think it's just
But then again, the other epidemic coronas were never in as many people as this.
Well, yeah, I mean, and probably
SARS-1 and MERS, right?
Yeah, right. They never really were able to transmit anyway.
Although the common colds are in many, many millions of people
right now. So the question is, what happened in 1890 or whatever?
It may have been like this for a while.
It may have been like this for a while, and we don't. I mean, it wasn't the same kind of society, so it probably wouldn't have quite the impact. People weren't flying all over the place, or it may have just been a lot more of a slow burn sort of spread because of transportation at the time.
I guess the hope is that this SARS-CoV-2 will eventually end up like a...
Yeah, that's my...
Because they do undergo variation.
Yeah, they still vary a lot, and there's different genotypes, and they'll select, and it's like influenza. It's not static by any means. They'll keep adjusting themselves, and hopefully that'll make life easier to manage, and we'll get vaccines that are a little better matched, and there'll be some drugs coming along, and hopefully more than we have right now, but. And then we'll
if you use the drugs early, they can save lives.
Yeah, they can, definitely.
They can. So do you think that the furin, the loop,
the loop,
which is cleaved by tempers 2, do you think that explains the difference between SARS-1 and 2?
I think the loop makes the difference, between and transmittability between SARS-1 and SARS-2.
Of course, we can't do that experiment. People would go crazy, right? But okay.
Right.
And so this SARS-CoV-2 is a virus that just randomly arose in bats and happened to have a furin cleavage site, and it got it, and then it happened to meet people, right?
Yeah, and it came through. That's I'm not entirely following the whole origin story, but I'm keeping an eye on it, and it's I don't think it came straight from bats to people. It came through some kind
other
of
animals,
but where and how that there was some intermediary somewhere, and the question is where and how, and it's obviously linked to that Wuhan market, but the details are still pretty mysterious.
But I think you wrote in one of your reviews that the furin cleavage site is a process of polymerase stuttering, right?
Well, that's what with influenza. So we have to be a little careful.
Right, that's with influenza.
Yeah, with influenza. Okay. So you get these run. This is what a polybasic site is. It's just a run of repeats.
Right, okay.
So with coronavirus, we don't get these big inserts like with them. It's not just suddenly putting in a polybasic site. It's adjusting itself more. It's a little bit more like H9 influenzas, actually, I think. Mm-hmm. So it's H5 and H7 do that slippage,
right?
And but 9 is more. It's more like a coronavirus. It's more a slow process of adjustment. And if you track H9 influenzas over the years, you'll see something kind of similar going on.
But if you compare, let's say the closest bat SARS-CoV-2 like viruses, there's a gap there's
a gap. Yeah.
So
right.
how would you Would that be a recombination event?
Probably some recombination event. Yeah, it's not. I'm getting outside of my territory in terms of expertise. But one thing, a term I learned over the pandemic is epistemic trespassing. Have you come across this?
Epistemic trespassing?
Yeah, this is.
What is that?
It means like being an expert in something you're not an expert in. Epistemic is your expertise, and you're trespassing into somebody else's expertise.
We call that having to stay in your lane, right? I think I know.
So that's the fancy term for that is epistemic trespassing.
Interesting. Well, that's happened a lot in the pandemic, right? Because everyone wants to be an expert. But
I think recombination is what's going. There's little chunks, if that's the right word, in indels.
We need to sample bats more, and we'll find. Although this may have happened already a couple of years ago, we may never see.
Right. But other species too. I think a lot of people. But I'm actually thinking that more about small terrestrial mammals like rodents, but also like shrews and hedgehogs. I think they're a good. That's an area that's not really well understood and studied, and there's some very interesting things going on in those small mammals. So I'm actually I'm moving out of my comfort zone in the lab and getting into the field a little bit. At least we have this new department of public and ecosystem health. Raina Pralright was on your show, right? A few weeks ago. So she's arrived. I'm got a joint appointment in that department, so I'm kind of going into that a little bit and thinking about wildlife sampling, here in my own lab, and sort of piecing that all together in terms of the biology of the spike, and I think there's a very interesting set of things to do there. But I'm sort of focusing on the shrews and the hedgehogs. And I'm told there's no hedgehogs. I'm from the UK, and I expect hedgehogs everywhere, but apparently no.
Here. Okay.
Not in North America. I'm learning some wildlife biology from my colleagues. With shrews, there's lots of shrews.
Yeah. So let's talk about feline coronavirus, which you're very interested in. So where do we start? What is feline coronavirus?
Is
it alpha coronavirus?
It's alpha coronavirus, right? So it's this weird alpha coronavirus, and you do the alignment. There's no S1 S2 loop except in feline coronavirus 1, but then there's feline coronavirus 2, which is more traditional. Let's say.
What are these, two serotypes? You would say.
Well, it starts out as two serotypes. I mean, that's what it was called for a long time, two serotypes. But I think it's way more than. I think it's just a completely different virus. I'm starting to call it a genotype now. I think I'm okay doing that. It's a different genotype.
Okay.
So there's very little sequence alignment. It's just, and the type 2 virus, by the way, is in cats is really a dog virus. So there's very clearly recombination in spike. So the history is pigs. So TGEV is transmissible gastroenteritis of pigs.
Mm-hmm.
That sort of got into dogs as canine coronavirus type 2, and then that recombined. So feline coronavirus 1 and canine coronavirus 2 recombined to create feline coronavirus 2. And with just the spike protein and the all 3 is just drop right over.
And feline 2 will infect dogs still?
Yeah, so it's not a major cause of disease generally, but it does infect dogs. So it's enteric. So a lot of these viruses in animals are enteric.
Mm-hmm.
And I think that I'm starting to come around to the idea that's because they're not cleaved at S1, S2. So they just make it inherently more stable. And you were here yesterday talking about polio, and we can't put a coronavirus in 1% SDS.
No.
So somehow it's got to survive through that through that GI tract if it's going to be an enteric virus. And I think that the alpha coronaviruses in animals are generally enteric, and so they're made a generically stable spike because they don't have an S1 S2 loop. But then the question then is how because feline coronavirus is so it causes FIP. For those of you who have cats, you may have heard of FIP, feline infectious peritonitis. Mm-hmm. It's a sort of really severe, horrible peritonitis. Well, classically peritonitis, so the pleural and the peritoneal cavity fill up with fluid, big vascular leakage, horrible disease. So that's the classic so-called wet form of FIP. And then there's a dry form which tends to be neurological. It goes in, gets signs in the eyes. It definitely gets into the brain. It's macrophage tropic. So the macrophages either trigger this inflammatory reaction or they don't. And if they don't, they just migrate and get into the brain eventually. Is the idea. But the idea was that in the field, and it was an internal mutation. which was there was FECV, feline enteric coronavirus, and FIPV, and what separated them was a so-called internal mutation.
Internal in the genome.
Internal into the cat in a way. So
it's in the cat genome or the viral genome? It's in the cat genome.
No, in the viral genome.
But it happens in the cat.
It's like the cat is like one big giant vero cell in a way.
So it gets multiply infected.
So the cat there's a form that transmits very readily, which is the so-called FECV, and then that mutates to become FIPV. But FIPV does not transmit. So
I see.
So it's not. Then we figured this out like many years ago. If you start sequencing FIPVs, they've got mutations in the furin cleavage site almost every time. So I think that tracks with this stability. So it can't. That's why it can't transmit very well, because it's back to SARS. It's kind of gone from SARS 2 to SARS 1.
What is the selection for that? Do you think make it more stable?
Probably make it more stable. Yeah. So the question is, how does that connect to infecting a macrophage? I don't know. So it's a mystery virus. There's lots of mysteries about this virus.
A big selection pressure is transmission, obviously, right? So it's funny that it would lose transmissibility.
It's losing so it's one of those. It's a virus accident. Clearly, it's a virus. The virus wants to be transmitting from cat to cat.
Yeah, of course.
Right, but then it makes this mistake and gets into macrophages and all.
And then you have a different disease, peritonitis.
That's when you get the disease. It's kind of.
So how do cats get it? From other cats only?
Yeah, it's just
so if you have
or maybe from. I think there's a lot of cat-dog interchange probably.
So if you have a cat, it's mostly at home inside. It's not going to get coronavirus.
If you look at the data, it's like every time. If there's more than 6 cats in a location, then it's going to have feline coronavirus. It's kind of like the calculation. When I talk to shelter medicine, right? So it's the problem is in an individual cat. The problem is all cats have come either through breeders or shelters. So even if the cat's in a house just all by itself, we have a cat at home all by itself, getting lonely during the day, like we're all in the office here, but my kids have left home. So that came from a shelter. So when it was in the shelter, it got exposed in that.
And so that cat could still have virus.
I think that cat could still have virus.
It's persistent.
I think it's persistent, and this is where I need to give credit to Raina, because when Raina was here, she was talking about bats, and she put up a slide from a paper she published a while back in PLOS NTD, neglected tropical diseases, and it was what she called the silly model. This is talk about epidemiology for a second. We got SIR models, right? And so she was the SILI model, silly, which was susceptible, infected, latent, infected. So this is what she thinks goes on in bats. The bats, maintain the virus persistently. And as a virologist, I would argue with the L term, and I've changed it to P to make it persistent and latent.
Because I think is what we think about herpes virus,
right? And I was trained in herpes viruses, so I immediately push back on
Makes
that.
Sense.
But SIPI, so it's a SIPI model, not the silly model, but the same concept. But I think that's what happens in the cats. They get exposed as kittens.
I see.
But then it goes into some reservoir, maybe in the GI tract, because the cats will intermittently shed virus, so it clearly goes systemic and it sits somewhere. That's part of the goal right now, is to figure out where in the cat that virus is hanging out. It may be the GI tract, it may be somewhere entirely different. We don't know. No.
And so, what is the trigger for it suddenly exacerbating?
Stress. So this is we've got some conjecture going on here, but we had a story again, shelter medicine. We work a lot with shelter medicine, and they had a house fire in the town next to Ithaca, and the cats had to escape the house fire and got picked up and taken to the shelter. 7 cats, and then 3 cats got FIP as they came into the shelter, and then cat 4 was pronounced fine and got adopted, and a week later got FIP. So there's 7 cats, 4 of them got FIP right away, and I think that's the stress. I mean, and one of the story that shelter medicine told me is that what you have to think about for a cat going to a shelter is just like a human being abducted by an alien. It's that level of stress. So cats get easily stressed, and it's that level of if they're taken from a home and get it's you be picked up by an alien and taken to another planet. That's the way we think.
Maybe some people would like to be taken to another planet. It wouldn't be a stressful thing, right?
But so cats, but being in a shelter is very stressful on the cat. And shelter medicine tell me it's not. We're in Tompkins County. We have a very well run shelter, and they get FIP, but it's not a huge problem. It's the shelters that don't have the resources to really manage their population that really have the FIP problem. It's becoming, and it's clearly becoming more as cats get into more and more dense housing. It's becoming more of a problem. It was it was picked up in the Cornell story is Jean Holtzworth, who was the 1950. We have a whole set of pictures on the corridor down here. She was like the only female graduate in 1950. It's completely different now. It's all predominantly female, but she was a pioneer in feline medicine, and
to veterinary school. You mean?
To veterinary school, yeah. So she actually was a teacher in. She had a PhD in Latin. She was like a college teacher teaching Latin, and then I just looked up her bio the other day. So her cat got panleukopenia, which is parvovirus in cats, and then that after that she said, I'm going to be a vet. This is going to be my so at some point around the age of late 20s, I think she just gave up being a college teacher in Latin and went off to came to Cornell to vet school, and then went to Boston and set up this, she was like a real pioneer in feline medicine back in stayed in Boston in practice. So she picked it up in Boston in the 1960s, but it was modest. It wasn't, but then as time's gone on, it's just become more and more of a problem. And I think that's because cats are getting into more and denser housing in general. It's just a lot more, large populations for the virus to transmit through, and it just builds.
So do you have any idea what fraction of cats, all the cats have it?
Well, if you go to a breeder or a shelter, pretty much every cat has it. Yeah. I mean, we've had situations. Actually, the whole story started with Ed Dubovy. I don't know if you know Ed Dubovy. He was our former head of diagnostic virology here at the vet school, so very well known in veterinary virology. And he came to me one day and he said he'd heard I was started to work on coronaviruses. So he said, I've got this breeder, and he sent me all these samples. I have no idea what to tell her. And like, can you help? So, because all the tests don't discriminate, it's just feline coronavirus. There's no way of knowing if it's the pathogenic or the non-pathogenic variant. So we started to sequence, and then right away, I remember thinking it was, I kind of had this sort of panic attack in my office because we started the sequence, and the sequence came in, and right in the S1, S2 junction, there was a furin cleavage site. And I was expecting it not to be there because it was an alpha coronavirus. So I thought, it's like the high path bird flu in cats. So that's what's happened. And it turned out that was we had to flip that idea altogether because that was the normal situation.
Right. And that's the not pathogenic.
So that's the non-pathogenic version is the one with the furin cleavage site. So it was entirely, the logic was, I had to flip the logic completely from HA, but this is what keeps me excited about science. So that's it's Ed set me on the track of, like, as soon as I saw that, I thought, wait a second, what's going on here? And then we started the sequence. Then we went to pathology, right? We had Gerald Duhamel, who's one of our pathologists, and he had a lot of FFPE tissue, of cats. So we were able to go into that formalin fixed tissue of FIP cats and sequence. And then that's when we saw that, okay, whatever we're seeing in the fecal sample in the live cats, even if it's an outbreak of FIP, it was like an outbreak of FIP in the shelter, in this breeder. And it was like almost every single time we saw a point mutant somewhere in that, and if you line up the biochemistry with the arginine spacing, it's clearly like one of those key arginines is gone. But it never eliminates it. This is what's kind of puzzling me now. It's because it's duplicated. And this is what biology does. If there's something really important, it puts things, it puts it in tandem. It happens a lot with glycosylation sites. They're always like two together, but only one is used. So it's like a backup. So there's a backup, and the sequence is SRRS, RRS. So it's like there's two furin sites back to back. So if one goes, the other one's still there. So
it's still cleavable.
It's still cleavable, but not as cleavable. I think it's just it's not eliminated cleavage, it's just down-regulated cleavage.
That makes it more pathogenic.
Yeah, it's a lot. If you try and think about the logic of all this, it kind of drives you crazy. And this virus has been driving me crazy for like the past 10 years.
So did you start to work on, are you the first person to work on this virus and find this furin site change?
Yeah, we were. I think we're the first sort of molecular virologists to kind of think about this. There's people, it's been studied But I think the putting that concept of the genome, the difference in the genome, first of all, between type 1 and type 2 is a major thing, and nobody really thought that was a major thing before. It was all considered one virus. It's really two very different viruses.
And do others work on it now? I presume so.
Some, yeah. There's still not a lot. It's still it's a very niche area, which is, gives us plenty of space in terms.
So I presume there's no vaccine, right?
That's yes and no. So this is one of those. This is where ADE kicks in, antibody dependent enhancement. This has come up with COVID, like, and so there are some examples where this happens, and it does seem to happen with FIP so that because it's infecting macrophages, if you get these so-called non-neutralizing antibodies, then that they will get recognized by FC receptors on macrophages, and it will actually drive infection into macrophages and make things worse.
Okay.
So there's some very famous papers from a lab in Utrecht, Peter Rotier's lab, and including others. It was the Netherlands is a gold mine of coronavirologists, so Peter Rotier and his colleagues worked for many years, and there's a paper. It's like early death following FIP vaccination. It's like you see that paper, and it's like, well, this isn't so there is a vaccine, but it's a type 2 virus, so you have to keep that in mind. Nobody's ever tried to make a type 1 virus vaccine. So we're actually starting to think about that now. Yeah.
So the cat gets sick. There's nothing you can do, right?
Again, yes and no. So until about 2 years ago, that would be the answer. So we've been trying to develop diagnostic tests, right? So we think, it's been a long, hard road, but if we can sequence that cleavage site, we can make a prediction at least of macrophage tropic or not. Because if we see the arginines disappear, we can say, okay, at least make a prediction. It's not yet black and white diagnostic, but it's useful. And I was always very concerned about false positives. So because if we had a false positive, that the vet will, the cat gets euthanized. That's the basic treatment strategy. If you've got an FIP cat, there's nothing you can do. You need to euthanize, and that's a big deal for the cat. Certainly a big deal for the owners. Yeah. So we want to eliminate false positives. So as absolutely as much as possible. And we had a cat that was came in. We were trying to collect. We had a healthy cat study, so we put out a call to get feces from healthy cats, or at least from non-FIP cats. And then right away we started to get these cats that had FIP sequences, and it's like, okay, this is not going to. This is a problem. It turns out that those cats, 5-10 years later, some of those cats have now died, and they've still got feline coronavirus in them. 5-10 years later, this is kind of what's making me think, this is really a lifelong infection in these cats. But they're dying of other reasons, but they've still got that virus there. But the FIP sequence has gone away. It's just now it's gone back to the so-called FECV, but it's in many. It can be in many tissues in the cat, in the eyes, and the spleen, and the liver. It can be in lots of different places. That's the thing. So back to but then now. There are these drugs, right? There are in cat world. There's a magic FIP cure, which is basically remdesivir. Remember remdesivir? Yeah. Right. It got tested. The lab in UC Davis, Niels Peterson, sort of did some clinical trials on cats with remdesivir. It's a number I forget. It's GS something with a big number. I always forget the number. But and it worked like a dream in these cats, in these FIP cats. He published a couple of small clinical trials, and now the cat world's gone, crazy for these things, and but it's not FDA approved. So
it has to be for cats.
Yeah, it still has to be FDA for a vet to use it. It has to go through FDA approval for an animal. But apparently it works great.
So
as an
does
FIP
it clear?
drug.
Does it clear?
Well, that's
virus.
That's what we don't know. I don't personally. I think not. The symptoms go away.
So it saves the cat's life, right?
It saves the cat's life, but whether those what we want to try and do now is go to those cats, those treated cats, and go and wait. We kind of have to wait for them. We have to wait for them to die for some other reason, and then we can go. Look at their tissues.
Sample.
We can't sample. If it's not shedding in the feces, then we can't really biopsy it or anything. It's that's the challenge of clinical trials in cats. We can't. If we had a lab animal, we could do it in a lab. We could certainly set up an FIP study in a lab, but don't want to do that. And that's ethically, that's not quite where I'm at. But
and there's no other lab animals.
There's no mouse model or anything, unfortunately. So it's cats or nothing. So we're pretty limited. We can't do cell culture. We can't do mice. We can't do. We have to do everything in cats. And ethically, we don't want to be doing a whole lot of
of course,
experimental challenges in cats. That's not, we want to avoid that. So we kind of go to the clinic and try and work with our clinical colleagues, work with shelter medicine, and get these animals from the clinic and do it that way. But now, so now it's all available through the internet. Let's say you can just go into a website and type in and self-diagnose your cat with FIP, which I have a problem with straight away, but that's just me. And then fill in, give you a credit card number, and they'll ship this remdesivir, and cats but it's all IV, right? So and it's not cheap. It's thousands of dollars.
Wow.
But people are willing to pay it, and the vets, but they can't. The vet can't
because it's not FDA approved.
Right. So what they've worked out a scheme whereby they'll train the owners to do the IV injections. They'll treat it as an outpatient and do it that way. So that there is an oral form now too, as well. But if you've ever had a cat, trying to give a cat a pill, I'd rather give a cat an IV injection than try to give it a pill. But because
oh my gosh,
you can get bit very easily. But anyway, so it's a very interesting scenario, because we've got, and in the UK it's actually been approved, and there are.
Okay,
in the
so
UK,
bring your cat to the UK.
You have to take your cat to the UK. And I'm actually going to a conference in Glasgow, like in a couple of weeks, that with a bbig ISCAID. It's the animal infectious disease meeting, and there's a lot of talks on FIP treatment there, and there's all the UK vets are able to to really do it right, and we're just waiting for the US to kind of catch up in a way. And so it's fascinating to me that this thing really works in cats, but it saves their life, right? Saves their lives, which is kind of what you want for but it's I do worry about antimicrobial resistance as monotherapy.
Sure.
It's not well regulated at all now. So I have two goals. One is to get better diagnosis, to really be able to figure out what's going on with these cats, and are they really cleared or are they not cleared, and do that, and also to start looking for new drug targets and ion channel inhibitors and other things. I think because that's where the ORF3 comes in. So this downstream gene from spike is the cat and dog viruses are very unusual in that ORF3, and they have an ORF3C, which is an ion channel, which is unique to cats and dogs, and that's a very good drug target, for instance. So,
does Paxlovid work? Is there an M protease? Right?
Yeah, it should work. I don't see why it wouldn't. And I'm not actually completely up to speed on one, but then on. I mean, they all. I mean, really, the RDRP inhibitors and the protease inhibitors should translate very nicely into animals. The spike is obviously different. The ORF3C is different, but most of the other genes are broadly similar. So they should cross that species barrier pretty nicely. So. Yeah, and cats that's why I kind of worked on. I'm in a vet school, and I was working on SARS. But thought, where can I make impact? And we have a feline health center.
Yeah, that's perfect.
And they came in and really funded as well, and they've been very supportive.
That's terrific
with doing this. So I'm kind of that's where I'm kind of making my impact and trying to get better diagnosis, trying to really completely rethink the whole. I'm kind of almost gone back to basics and almost like throwing away all the textbook and starting from scratch and saying, okay, if this were a brand new virus, this is what we would do, and just sort of rethink the whole thing, because there's a lot of people getting mixed up between type 1 and type 2, and that's not most people don't discriminate there, for instance. That confuses everything straight away. Well,
it's an interesting story, but also cat lovers will be grateful, right?
We have no shortage of cat lovers who write in and donate them. Yeah, so anybody out there who's donated a cat or a sample to us, thank you. The labs are remodeled, but very soon we'll be back to be able to handle bodies of cats and be able to go do the necropsies, look at the tissues, and really do the sequencing. Finally made we were doing Sanger sequencing for a very long time, and I'm not much of a techie, so now I have a postdoc who's running nanopore and Illumina, and we've got colleagues up at the Baker Institute with Laura Goodman up there, who's has been very helpful.
Nice.
So I think we're in the 21st century with our sequencing finally now, and we piece together the clinical and the molecular, and really in that, I think what it, to me, is a really fascinating little sweet spot of coronavirus biology in cats. And it's good, but it's just getting like with many things, it's getting funding. We have to convince the NIH that cats are cool.
Well, maybe cat lovers can help fund you as well.
Contact the Feline Health Center. They're more than happy to talk to you about that kind of thing.
All right, we'll put a link in the show notes to the Feline Health Center here at Cornell, and if you want to support Gary's research, you can contact them.
Yeah.
All right. Fascinating. Thank you. That was great. That's special TWiV here in Ithaca. You can find the show notes at microbe.tv/TWiV. Send questions or comments to twiv@microbe.tv. And if you have a question for Gary, I'll forward it to him. If you enjoy what we do, we'd love to have your support. You can go to microbe.tv/contribute. Gary Whittaker is at Cornell University at the School of Veterinary Medicine. Thanks for talking.
Welcome.
Appreciate
Been a pleasure.
it again. And I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWiV, Jolene for the timestamps, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.